Hello, good evening. So it's really a pleasure for me to give the second talk this evening, especially as we're staying right on topic and staying down in the ocean, because tonight I wanted to talk to you about what I titled creatively, "To See or Not to Be Seen," which is about how marine fish actually adapt to the very difficult visual environment in which they live. So what do I mean by difficult visual environment? Well, actually the lighting conditions in the ocean are very different from that what we're used to here above the sea. And this is mainly because of two different factors. The first is light intensity, and you can really nicely see this here. So at the sea level, if we set this to 100% light, the intensity of the light gets low very, very quickly. So at 30 meters, most of the light has actually already been lost. And everybody who's ever scuba dived will notice that if you go down, it gets dim really, really quickly. But this light scattering actually affects the different colors of light to different extents, and which we can see here. So this is the second factor. So red light, because it's scattered more than any other color of light, doesn't penetrate the water at all, almost at all. And so in the deep sea, you only essentially have blue and a bit of green light. Which means that this is why all these deep sea pictures have this blue-green tinge to it. So this is a very tricky visual environment, and marine animals have found numerous ways to actually adapt to this. And I want to explain some of these strategies of adaptation using two examples. And I picked these examples mainly because the crappy weather outside is kind of putting me in the mood for a bit of a holiday, at least on a mental space. And so we're actually going to go here to the Southern Pacific, where right now it's like super nice weather, and I think most of us would prefer to actually be maybe here on a sunny island somewhere in the Pacific Ocean. This is those of us who aren't as lucky as Steffi, who actually got to go there for her job. So we just have to go on like a mental holiday. And the first fish that we'll visit are these very pretty coral reef fish, which are called fairy wrasses. And they live somewhere between 10 and 30 meters below the sea level. So you still have quite a bit of light, but all the light is essentially blue or greenish. And so there's no red light at this depth. And this means that most of the fish who live down there have lost the pigments in their eyes to see the red light, because, why invest in making pigments if there's no red light to see? But actually, it's been known for a while that the fairy wrasse have these red light pigments, and why they do becomes clear when we look at these fish in more detail. So this is what the fish looks like under white light. So if you keep this in an aquarium, this is what you'll see. But if you just illuminate them with blue light, you see a stunning difference. So these fish actually produce red fluorescence, and this very distinct pattern all across their body. And the role of this for their behavior is really important. So this patterning is really important for their behavior. And you can really nicely show this by doing some experiments in which you put the fish in front of a mirror. And because the fish is relatively stupid, it doesn't recognize itself in the mirror. So it thinks it's other male fish, because only males have this patterning. And so it thinks it's a guy who's trying to take his territory away. So he'll show, if you see his mirror image there, he'll show very aggressive behavior. But then if you put a red filter in front of that mirror, this is what the fish looks like all of a sudden. So all the red fluorescence is gone. And this means to the fish, oh, I don't care about that other fish anymore that isn't fluorescent, because, well, he's a lower ranking male, so I don't have to display any aggressive behavior. Which means that these fish essentially have a secret language channel using this red light, in which they can communicate with each other and communicate their social ranking. And it's also important in attracting mating partners for these fish, because the fish that have the best fluorescence get the most girls. So as it works apparently on this deep sea world, I'm not sure, maybe the guys should think about adopting some red fluorescence. So these fish are kind of like the spy fish of the reef, which have this secret language channel, which they use because none of the other fish, so all the predators around them, can't see the red light. So their secret information is safe. And so this is the first example that I wanted to tell you about. And the second example is actually from this almost similar region, but specifically from the southern Tasmanian Sea, which is right between Australia and New Zealand. And there we're going to dive down much deeper and visit a fish called the glasshead barreleye. And they're these very strange looking deep sea creatures. And they live, and you can figure out why they look so strange, because they live 1,000 meters below the sea level. And usually everything you find down there looks pretty strange. And down there, there's almost no more light at all. And what that means is that most of the fish that live down there kind of drop the whole idea of eyes completely, because it's not really much use to invest in making an eye if there's not light around to see. But these glass-headed barreleyes actually have taken the exact opposite path. They've invested everything into their eyes to get as much of the little light that there is, get as much information out of it as possible. And what they use for their advantage is that the light that you get down there is coming all from one direction, so it's essentially all coming from above. And so these barrel eyes essentially only look towards the sea, the top of the sea, and see all the fish that are swimming above them, kind of like shadows that take away their light. And so their main visual system is focused towards the top, and this is what you can see here. It's kind of cut off a bit, but you can imagine. So the name is really illustrative here. So the whole head is essentially see-through. So it's one of those really modern submarine robots. And then it has this barrel-shaped eye here. It has this gigantic lens, which essentially is like a telescope that's pointing towards the top and getting as much light as possible so that it can actually see something. But at the same time, it makes sense for this fish to look to the top, because this is where all the natural sunlight comes from. But at the same time, down there, there's quite a lot of bioluminescence going on, which means these are animals that by themselves produce light. And so this can be some sort of plankton, or like here this kind of jellyfish. And this is this anglerfish, which is probably like the most famous creepy deep sea creature. And so these barrel-eyed fish, if they're only looking above, they'll miss all this bioluminescence around them. And so what is the poor fish to do? Well, the barrel-eyed fish simply developed a second pair of eyes, which I think is a quite unique evolutionary strategy, because most of us only have 2 eyes and not 4. So what the barrel-eye does is it has the big tube eye towards the top, and then it has a side eye to see all the bioluminescence around it. And here you can see actually like a dead specimen of a barrel-eye. And this is the top, this is the tube eye to the top, and this is the side eye here. And then closer you can actually see that this is 2 completely distinct two distinct eyes, and you have like 1 pair on each side of the head, so it's 4 in total. And if we look at this closer, this is a histological section. You can see really this huge eye here, and it has this big lens, and it collects all the light from around. It focuses on the retina here. So this is super efficient, using all the light it can get. And this is the side eye here, which is also evolutionary very interesting, because it arises from a completely different tissue than the main eye. And so this is developing essentially like the same structure through a different pathway. And in this side eye, actually, the retina is here on this side. So if you imagine the fish head is here, this is kind of like, it seems to be on the wrong side, because all the light will come in through this small opening here, and for it to get to the retina, it actually has to be reflected. And as it turns out, if you actually do computer models of this, this is the optimal setup to get light from as wide an angle as possible and still be able to focus it. But what this means is you have to reflect the light here somewhere. So essentially, you need a mirror inside a fish. And that's quite a challenge. So the main issue is, how do you make a mirror inside a fish? Well, what these fish do is they take a molecule that's very common in biology and in all organisms, which is called guanine, which is a precursor of one of the components of DNA, and they just produce tons of it, like literally so much that it crystallizes. And because we can also make guanine crystals in the lab, but they're really small and really crappy. But these fish, they grow these crystals very, very slowly over a long time. And so this is what they look like. They're quite big, so they're like up to 0.5 centimeter in diameter, these little chips of crystal. and they have this perfect mother-of-pearl sheen. And you can actually see here in the image how this crystal reflects the light of the camera. And then these fish have a lot of these little crystals placed on top of each other, so it's kind of like a disco ball that takes the light from a lot of different angles and reflects it onto the retina. And so this is how this particular species of fish solved the issue of getting all the light that is around there in the most efficient way possible. And I think with this I'm at the end of my talk, and I just think this is a very nice illustration of something that Steffi already told us, that in the marine environment we have such a huge diversity of which we have essentially no idea. And there will be so many amazing evolutionary adaptations to a lot of problems we can't even imagine from our very terrestrial perspective. And I think there will be a lot of very interesting discoveries to make in the next few years, because both of these species actually were described in their visual systems only in the last 2 years. So there's a lot of very new, interesting research coming from this marine biology area. And with this, I want to thank you for your attention and hope you have a nice evening.
Thank you very much, Johanna. I was really impressed by the size of this fish. It made me really startled. But I hope that people will discover even uglier creatures. Please, does anyone have a question? There is in the back.
Thank you, step out of the spotlight and anxious.
I'm actually very loud. Yeah, are there evolutionary constraints that they had to overcome in order to have these two separate retinas? That seems like a really hard tissue to develop. Are the retinas developed from the same precursor tissue?
No, so it's actually very interesting, because there's so far know that there's 2 species of this barrel-eyed family, and there it's known that they develop, their eyes look very, very similar, but they've actually developed this through different pathways, because the tissue from which these eyes originate are different in these 2 species. So there's 2 species which are very closely related, but which have independently evolved this side eye solution to their problem. And so I think, of course there's evolutionary constraints, but it seems to be quite an efficient way of solving, like getting the most information out of your environment. So I would look at it in the way of it's worth the evolutionary investment of making a second pair of eyes.
Cool.
Does anyone have more questions?
Hi. You said that the guanine mirror that these fish have develops over time. Mm-hmm. Does that mean that they are blind for a period of time in the early life? Or?
Yes, so that's the implication. So they will still have their main barrel eye, which is what most of the species in this family only have, this barrel eye. So that seems to be fine for surviving. And the side eye then develops over life, because it's kind of an outgrowth of the skin at the side of the head. And so this develops over time, so this is kind of an adaptation maybe to adulthood. But the main problem is that there's very few samples of these animals, because they're from so deep down. So I think there's been 2 fish described. Like, human beings have seen 2 of these fish. And now we're kind of concluding that this is how this works. But so it's really difficult to tell how this develops, so we just need more animals essentially.
There's one more question, but I have one as well, a very short one. If there are only 2 of these exemplars, how do we know that it's not a random thing?
Yeah, so there's 2 of each of those 2 species, so they're very closely related. And, it's really strange. It's a really strange tissue, and you would probably not expect this to occur in these 2 species so closely related independently of each other, just randomly.
Thank you. There's one more question.
Hi. I'm very interested about this, the fish having 2 pairs of eyes. How does the brain comprehend it?
Yeah, that's the perfect question, but we have absolutely no idea how this works, because you can't. So the fish are dead when you get the samples. So you can't do any sort of wiring, testing of the electrophysiology of the brain or anything. So we would have to catch live samples, but I guess that's pretty tricky.
Thanks.
We have time for one more question. Please.
Hi. I have a question. Hello?
Yeah.
Hi, I have a question. What is the practical implication of this research? I mean, you are doing, yeah, I mean, you are doing this research, fine, but is it using somewhere in practical?
Well, I think in general it's very interesting as like a basic evolutionary research to understand like how you can find like what sort of pathways life finds to solve the challenges. And I think you can never like learn too much.
No, I mean,
But I mean,
you
it's
use
not-
something it for the in human
Yeah, no,
research,
no.
for
This
human
is-
brain research, for example, or whatever. I don't know.
We can't really claim any of this sort of thing. It's really about curiosity and trying to understand like how different species adapt to their environment. And I mean, this you can make all sorts of implications. This has for environment change, the importance of the oceans. But I think it's really about trying to just get a grasp of the diversity of life, especially in the oceans where we have like only a very like tiny viewpoint of how the real diversity is there.
Okay, thank you.
Let's thank Johanna for an awesome talk.